Hello everyone, in this video we will talk about non-viral method of gene therapy. I have a separate video on viral method of gene therapy. If you want to know about viral method of gene therapy, please go through my video. Today we will discuss what are the different methods, most commonly used method of non-viral gene therapy. Okay, so number one is your microinjection. Microinjection is a process in which the genetic material is directly injected through a syringe, which contain a needle, and without any carrier. It is the simplest and safest method, you can say, simplest and safest, because there is no carrier. You are inserting the gene, only the gene, nothing else, no chemical particle, no virus, nothing. So this is very safe, but the problem is that the efficiency is low in this method, because there is high chance of getting degraded by nuclease enzyme. The gene might get degraded by nuclease enzyme, and it might be cleared by mononuclear phagocytic system. It's where there are huge chances of getting degraded. So this method its efficiency is low, you can see. The attractive candidate tissue for microinjection are skin, then liver, cardiac muscle, solid tumors, etc. Normally performed under a specialized optical microscope. And the setup is called micromanipulator. So this was about microinjection. The next commonly used method is gene gun. This method was first used as a gene transfer technique in plants. This was first used in plants. The method is based on the principle of delivering DNA molecules. They will be delivered, they will deliver DNA which are coated with heavy metal particles like gold, silver, or tungsten. This is done with the help of this kind of gun. They are inserted inside this gun, and DNA molecule coated with this heavy metal particle are delivered by crossing tissue at a certain speed, and the sufficient speed is achieved by high voltage, electric discharge, spark discharge, or helium pressure discharge. Next comes your electroporation method. An electric current is applied across a cell membrane. Suppose this is a cell, electric current is applied. Then what will happen? There will be pore formation. Here pore will be formed in the cell membrane. These are temporary. There will be temporary pore formation, and due to formation of pores, there will be uptake of exogenous molecule. Whatever exogenous molecules will be there will be taken up by the cell through the cell membrane. Such as your DNA will be there. The solution, if contain DNA will be taken up by the cell then it will go to the it will be imported into the nucleus. The method is like that. Due to the generation of electric field, pores will be formed, and through this pore DNA will be taken up inside the cell. There is another method called sonoporation. Where ultrasound is used to temporarily permealize the cell membrane to allow cellular uptake of DNA. Ultrasound is used. There is another method called photoporation. Where laser pulse, single laser pulse is used to generate transient pores. Then comes the most common method called lipoplexis, where liposomes are used to deliver the gene of interest. Plasmid DNA can be covered by lipids into organized structure, such as, suppose the structure will be similar to a cell membrane, where there will be hydrophilic region inside and hydrophobic regions outside. Outside will be hydrophobic and inside is hydrophilic. Just like the cell membrane. Okay, and inside your DNA will be inserted, gene of interest. So this is the structure. There will be positive charges around it. And this, it will form, sorry about that, as DNA. Okay, your DNA. These are lipophilic structure. Your DNA will be added and it will form a mycelium like this. Your DNA will be inside and a lipoplex will be formed. Okay, it's like DNA covered with lipids in an organized structure. Okay, the complex of DNA with lipid is called lipoplexes. The positive charges of liposomes allows interaction between negatively charged cell membrane. So when it is allowed to interact between a cell with a cell, then the cell membrane, the negative charges on the cell membrane will interact with the positive charges around the lipoplexes, and thus penetration into the cell is permitted. Suppose this is your cell, has minus charges, both will interact and therefore pores will be formed through which your liposome will be inserted inside the cell. Somewhat like this, the formation. Suppose this is your cell and suppose this is your liposome. Plus, minus. So the cell will be engulfed, the liposomes will be engulfed by the cell membrane. Something like that will happen. Okay. So there are possibilities that there are different possibilities whenever the liposomes get inserted inside the cell through the plasma membrane. It may be either transported to the nucleus. Suppose this is your nucleus with nuclear pores. Okay. Or it might get degraded. If the liposome is released early in the cytoplasm, then what will happen? The DNA present inside will be released and it will get degraded by various enzymes, endonuclease enzymes present in the cytoplasm. Okay, or it might be transported. Okay, suppose this is the structure. So transported. During transportation, it might get degraded by lysosome. Okay. If lysosome comes on the way, it might degrade the lipoplexes. Lysosome mediated degradation. Or it might get transported near the nucleus, or when it reaches near the nucleus, the DNA will be released. Okay. Then again, one chances is that it might get infused with the what it is called endoplasmic reticulum, which might lead to dissociation of the DNA molecule. Or it might get transported through the nuclear pore. with the help of NLS proteins. Suppose these are your NLS protein. It will give signals for transportation of the DNA to the nucleus. So your DNA will enter the nucleus. Now it will bind to the genome of the target cell, and this is how this transfer of the gene is done with the help of lipoplexis. Even though this gene therapy process have very high potential of curing diseases at genetic level, it's still in the clinical trial processes because of some ethical issues, okay, and risk, lot of risk is involved. So we will see what are the ethical issues related to this gene therapy method. Gene therapy method involves making changes to the body's set of basic instruction, right? Like gene, it raises many unique ethical concerns, like will the high cost of gene therapy makes it available only to the wealthy, because everyone will not be able to take help of this treatment if people don't have a lot of money. So this is one of the ethical issues. Then there is another question: could the widespread use of gene therapy make society less accepting of people, of people who are different? Should people be allowed to use gene therapy to make society less accepting of people who are different? That's another factor. And then there will be who will have access to this therapy, okay, who will know what to do, what not to do. So this is a big question. Then should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence? Because if this is this becomes a common process, then people may want to be perfect. They want to have a good height, they might increase their intelligence, they might want to have athletic ability and many more. Okay, people just want to be perfect. So this might pose many ethical issues. These are the concerns raised by various people for gene therapy. So I have I have made 3 different videos regarding gene therapy. Please go through my videos to understand about gene therapy, what are its advantages, disadvantages, what types of gene therapy, what are the vectors used, whether viral or non-viral, everything is included in these 3 videos. Thank you for watching.